This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 609, recorded on May 1st, 2020. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Good afternoon, Vincent.
It's a great day today, isn't it?
Yeah, it was a little bit more intense yesterday. Too bad we didn't have yesterday's because the rain was almost coming into my apartment. It was absolutely elemental outside. I almost looked for funnel clouds.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. I'm surrounded by elements too, but it's certainly less rainy today than it was yesterday. It's supposed
Elements.
to be gorgeous weekend.
Elephants? Did you say elephants?
Yes.
From Austin, Texas, Rich Condit.
Hello, everybody.
Hello,
We
Rich.
have another glorious day here in Austin, 84 degrees, headed for 89. So we're at 29C and a beautiful sunny day, light breeze. It's wonderful.
From Southeastern Michigan, Kathy Spindler.
Hi, everybody. It's bright and sunny, and it's 61 Fahrenheit, and you all know what that means in Celsius. It's 16.
16.
You're under 16, you bet. From Madison, New Jersey, Brianne Barker.
Hi, everybody. I'm pretty happy that it's not as rainy as it was yesterday. It's still pretty gray here, though. Clouds don't look great. And it's also 61 here.
We
Right.
have a full house. Everybody's here.
It's palindromic.
Plus, we have a guest today. She is from the city of brotherly love, Philadelphia, University of Pennsylvania, Perelman School of Medicine. Susan Weiss, welcome back.
Thank you. And actually, I see pretty blue sky with white puffy clouds, and it's 66 in here in the south.
You were last on TWiV 460, which is an episode we did down there. Do you remember?
Yes, I remember it well. It was fun.
It was a lot of fun. I wanted to ask you, do you like Philadelphia? Is it a good city?
Well,
This is being recorded.
After living here for 40 years,
Wow.
I do like it now. I didn't like it when I first arrived here, but I like it. I'm from your city. I'm from New York originally.
What part of New York were you born
I
in?
was born on Upper West Side of Manhattan, and I grew up in Yonkers.
Oh, wow. You still have that accent, don't you?
Not that much. Not as much as some people.
Well, that's for sure. But you spent some years in San Francisco, right?
I did, yeah.
Do you like that a lot? Do you like that city?
Yes, that's my favorite place. In fact, I just came back from 3 months in sabbatical in Southern California, which I really, really liked. La Jolla.
Oh, that's a
Ooh.
wonderful place.
Yep.
Nice.
La Jolla is pretty nice.
We just got there under the wire before all this shut down.
Oh, so you were there during the shutdown beginning?
No. We came back just before.
No.
Yeah.
Might have been good to be shut down there, right?
Maybe.
All right. So if people want to know more about you, I think we got your history last time on TWiV. We all watched your video, so thanks for sending that.
Yeah, that was
It's an
great.
outstanding video.
Which one was that? I've been to so many. Which
One
one?
with the basic coronavirus data. It was fantastic.
Symposium, maybe. Yeah.
Right.
Yeah, it was the symposium, yeah,
Perfect.
Which-
It was perfect.
which I think had people like Paul Bates, because you kept shouting them out. And
That's
Stanley
right.
Perlman was coming up
Yeah,
then.
yeah.
Perlman.
Yeah, that's right.
Yeah.
That's right.
Yeah.
Yeah, and
It was
for,
really good.
for listeners, we'll have the link in the show notes. And Susan's video is a really great overview if you want to
Here.
understand the biology and also the history of coronaviruses.
I also read your JCI article, right? 40
No, it was
For-
JEM.
JEM.
Yeah.
Close, right?
Yeah.
40 years with coronaviruses. I
Right.
like that title. And I wanted to ask, before we talk about the current situation, you were working with Harold Varmus, right, on retroviruses.
Well, actually, with Bishop and Varmus. I was really, in that lab, you were either a Bishop or a Varmus postdoc, even though you were kind of mentored by both of them. I was hired by Mike. Yeah.
But you didn't want to continue retrovirology.
Right. Because, if you're interested, why at the time, it was just before HIV. When I left, there were just people were starting to get sick, but certainly the virus hadn't been identified. And so the field was really mostly oncogene oriented. And I wanted to work on viruses, not on oncogenes. So that's why I switched.
And why did you pick coronaviruses?
A kind of funny reason, because I had done paramyxoviruses in grad school, and I liked working on like lytic RNA viruses. So I literally opened up Journal of Virology looking for something interesting, and I found this kind of sounds ridiculous. I found coronaviruses because what was interesting about them was that it was just starting to be able to do molecular biology on them. I was really a molecular biologist at the time, mostly, and you could grow the viruses and you could do things with them. And they also had animal models. So I thought this is a great field, and hardly anybody worked on it at the time.
Perfect.
One thing I noticed in your article, Susan, that I could relate to myself is that you said you appreciated the great gift of time that Mike Bishop gave you to start your own future research while still in his laboratory. And I had the same thing happen when I was in Arnie Burke's lab at UCLA. He allowed me to start working on my future stuff. And that was really, you're right, just a great gift to have that.
Gift. And I don't know that I appreciated it as much, but just this week, my postdoc who just got a tenure track job asked me, would it be okay with you if I, gather data from my own lab while I'm here? And, how could I say no?
Right. Yes, right. You couldn't.
That's right.
Susan, actually, I have a question about basic coronavirology that came up during your talk. I'm not all the way through your video, but it's riveting. I'm learning stuff that subtleties that I didn't know, and I didn't realize that the subgenomic messenger RNAs were actually made from subgenomic negative sense RNAs.
Yes, isn't that curious?
Right.
Well, it strikes me, teleologically that provides an opportunity for amplification of them. That's kind of what I'm thinking anyway.
Amplification of the plus strands of the message.
Yes, exactly.
Yeah.
But I want to know who figured that out. Holy cow. Is it a bunch of people or focused on...
I'm trying to remember. I don't want to give credit in the wrong place or take it away. Well, initially, the history was that the idea was that the leader sequence that's on the 5' end of the message, that actually was transcribed, and that would be like a primer and would prime RNA synthesis off. There would be one long minus strand, and then this plus strand leader would prime at each intergenic sequence and make the messages. So that was the model.
Yeah, well, if I were God, that's what I would do.
Right,
but.
And that model was from Michael Lai and Steve Stallman originally, and then over the years it became clear. So I think some people that were involved in that was Stanley and Thea Sawicki. I think Ralph Barrick was part of that, maybe. Yeah. And David Bryan. David Bryan might have been the first one to show the multiple minus strands, and that would have been with BCV or one of the other viruses, not with MHV.
Well, it's fascinating.
Yeah, it is. Yeah.
So, Susan, the virus that caught your attention was mouse hepatitis virus. Is that right?
Yeah. But actually, in the beginning, when I started my own lab, I worked on MHV so the other thing that happened to me was I met Julian Leibowitz, who still works on coronaviruses too. And he was at UCSD, and I contacted him, and I went down there, and he gave me all his viruses and all his cells. This is a good story too. And then so we started working together, and he came up to to San Francisco for a week. And he slept in a tent in my backyard because he was allergic to my cats. And we worked for, we didn't have money in those days. We didn't go to hotels. So and then he worked in the lab with me in Mike and Harold's lab for about a week. And we set up in vitro translation of the mRNAs. And we worked together for that year or so before I left Mike's lab. And then, so we continued to work on and off since then. But then when I got to Philly, But
he was only in the tent for a week. You worked together at a distance.
At a distance. I went down to San Diego, and he came up to Philly. He was at UCSD, and he's now at Texas A&M. We're still good friends. And then, so then when I went to Philly, I decided I wanted to work on the human viruses because there was very little work on the human, on 2290 and OC43. So I got those viruses. In fact, as an aside, I just grew them up now after about 30 years. But anyway, and they were really difficult to work with. They didn't plaque well. They didn't grow well. They didn't have an animal model. So I sort of just put them aside and started working on the mouse virus again.
Can you tell me, what is the origin of MHV? It came from a mouse somewhere, right?
It was isolated the year I was born, and it was isolated so I won't tell you what that is. I'm probably older than everybody. And it was isolated, I think, from a paralyzed mouse, I believe. Well, there were different strains. One, A59 and JHM were isolated separately.
Okay. And this was in a colony, I guess, somewhere, right?
I don't know. I don't remember. And it was called JHM after someone, their names. And then of course, there are all these endemic mouse hepatitis viruses in animal colonies. They're GI viruses, and nobody really works on them. I think, people are afraid of the ones I work on, but those are even worse. I mean, for animal colonies. Colonies. They
can wreak havoc on mouse colonies, right?
Yep. Some places don't let you work on MHV because of that, which is dumb because the viruses we work on, you have to inject them.
So MHV is transmitted how among mice?
Well, it would be oral fecal for those strains in the, the endemic ones. But for the strains we work with, they really don't transmit unless you inject them. Yeah.
But in wild mice, are there MHVs or some kind of coronaviruses?
Probably. I would guess there are, just like in the mouse colonies. There are also rat coronaviruses.
But probably no one has looked very much, right?
I don't think people have looked that much in wild animals, in wild mice.
I mean, that's a big problem in general. Hardly anyone does wildlife sampling.
Yep.
I'm thinking that I'm just gonna throw this out there. I think that may change in the future.
Yes.
Oh, do you know whose funding was just whose funding was just revoked?
Yeah,
that's awful.
Isn't that horrible?
I think
EcoHealth Alliance. So, Susan, you must know the best.
Yes.
It was to do bat sampling, right?
I heard him give a talk recently or present data, really interesting, finding all kinds of SARS-like viruses in bats in southern China and Southeast Asia. Really important, I think.
And
it's extremely important.
And why was their funding suspended?
The ostensive probably politically, but I think the reason they claimed he was giving money to the lab in China, but he said that wasn't true.
No, it is strictly political.
Yeah.
The NIH, the reporters from Science tried to contact and get details on this, and nobody is saying anything except just, there were concerns. And of course there weren't.
No, I don't understand it.
Oh, I understand it. It's just depressing.
I'm experiencing a cytokine storm as I listen to this.
We got some anti-IL-6 receptor monoclonals for you, Rich. So, Susan, you probably heard that, Trump says, I have evidence that this came from a Wuhan lab, right?
Oh, and he said it before, hasn't he? Oh, yeah, he did.
Times.
He didn't say what it was, right? He wouldn't say.
No,
There isn't any, of course, but early on in this, you co-authored a paper on this whole issue, right?
Yeah. I was not the main author there, but yes.
So did they ask you to join and contribute your...
Yes. And I agreed with them.
Yeah. And you probably still agree, right?
Yes. I mean, it's certainly not man-made. Or human-made. That's ridiculous. That's really ridiculous.
Correct. We're not that smart.
That's what I said. Nobody could really. How could anyone figure out how to design a diabolical virus?
Exactly.
Seriously, it's so close to SARS but it's different all across the genome. So it's clearly not made from something else.
Well, and it's different in ways that nobody would have thought of based on our knowledge before this. Right. And there's just no credible way.
So there's no evidence that any virus has ever been let loose like this from a laboratory.
Of course not.
How stupid to start with this one. I mean, it's just an absolute idiot.
Well, but there's two issues, whether it's human-made and whether it escaped from a lab. They're two separate issues, really. Right. Related but separate.
Right, but I think so the escape from a lab hypothesis is only marginally easier to stomach. I mean, it really you would have to claim that the Wuhan lab, since they're the ones being accused, had a much more closely related SARS-like virus in their files than the one that's been disclosed in bats. And it is just, it's really, really a stretch to say that when there's a perfectly simple explanation based on what happened with SARS-1, based on what happened with MERS. I mean, we know coronaviruses do exactly this, and here's one that did exactly this. So the simplest hypothesis is best.
That's right.
Yeah, but you're not running for president, Alan.
Yeah.
And Dr. Shi, Zhengli Shi, is a really well-respected scientist. I just would not expect that from her lab.
No, that's not, yeah.
So have you met her at all?
I have. Yeah. I was at a meeting in Wuhan a few years ago. She hosted all the coronavirus people.
Cool.
Yeah.
Yeah, I understand there's going to be a virology meeting there next year.
That's what I heard. Yeah.
I'd like to go. I was in Singapore in December, and some people said, we'll invite you to Wuhan next year. So I would really like to go.
Oh, yeah.
We should do a TWiV there.
TWiV with Zheng Li. That would be great.
Yep. I have.
That's very cool.
Yeah, I have a former postdoc there also, and he just sent me some masks because I guess he didn't need them anymore.
Does anybody know what Wuhan used to be called by the Chinese?
What?
The City of Lakes.
No, I don't remember that.
It has 137 lakes, and then in parentheses, only 50 of them are still around. But
actually, I remember we did have dinner on a lake one night. It was really nice.
Right.
Susan, you so this SARS-CoV-2 is very similar to this bad isolate from 2013. So what do you think is a likely scenario for how it ended up in people?
I don't think it's the precursor because it doesn't have the infamous furin site, and also the receptor binding domain is somewhat different. So I don't think it's the immediate precursor. I think there's some other bat virus. I think the furin site is interesting, not so much for the pathogenesis, but for identifying the parent virus or the precursor virus.
Mm-hmm.
Yeah.
Remind me, does the pangolin isolate have the furin site?
No,
it doesn't.
And the pangolin virus is most likely closely related in the receptor binding domain. So that's why it was of interest. But the rest of the spike is less related than the one from the bat virus. So really, neither one of those viruses looks like an immediate precursor.
So really, if we wanted to get at the origins of this, we should fund more research on isolating that coronavirus.
Exactly.
How ridiculous. This is
I know.
Yeah.
So, Susan, there's another bad isolate, which was just published last year from a different group, not the Institute of Virology, where I think the whole 1AB region is the highest homology to SARS-CoV-2. Have you seen that one?
No, I haven't. Is that new? I mean, the newly published?
No, it's 2019. Eddie Holmes sent it to me
a
couple of weeks ago.
Why isn't that one in the news all the time?
Right. That's what he said to me. Why aren't people looking at this? They're always picking on the Institute of Virology, but they didn't even have this isolate.
But what about the rest of it? Is the rest of it similar to the SARS-CoV-2 or just?
It's more diverged. It's more diverged than RATG13, in fact.
I mean, it could be a recombinant, right? Because these viruses, because of that weird way it makes its RNAs, it's really prone to recombination. Because the polymerase has to jump just in its normal transcription.
Right.
So if you infect cells with two coronaviruses, it's easy to get recombinants out?
Yes. But someone was asking me this the other day, and I don't know the answer, how closely related they have to be. Mm-hmm. Probably pretty close.
For people that are listening to all this, not following the jargon necessarily, but still wondering, why do bats, of all animal species that you have to pick, why are bats? Well, Peter Daszak and I were at one time pretty close friends, and I learned from him that over 20% of all the mammals are bats. Mm-hmm. And it's the only mammal, as far as I know, that can fly. So you've got all these flying mammals going everywhere, except for humans, of course. But it's no wonder that all these animals. I wonder how many species of bats in China. I wonder where their paths cross on their migratory routes, and whether or not there's some chance of spillover from one bat species to another, and maybe that's how these things get passed around.
I'm not sure about that because different bat species seem to have different viruses in them. Like we don't have SARS-like viruses in North American bats at all.
Do you think they're susceptible, or just because they haven't been exposed?
I don't know. I'm just thinking it could be both.
But that's interesting that you see different virus species in different bats, even though I know you can get multiple species of bat in a hibernaculum. They'll be in the same cave, and they tend to be close to each other, but the viruses remain isolated within individual species.
I don't know about that actually. I don't know if it's proximity or they really are different. Like the MERS.
The rabies virus seems to spill over into different species.
Yeah. Well, some are going to be more species specific than others. Like the MERS bats are the viruses that the bats that have MERS-like viruses are different from the bats that have SARS-like virus, for example. Right. But that could be distance also.
And the other thing that's so interesting, of course, about this, Dixon, in terms of why bats, is that bats, because of their lifestyle flying, seem to have some very different immune responses. And I love reading about bat immunity. It's so interesting.
So I think there's a lot of speculation about that. I mean, it's probably different, but nobody really understands it. Like there's a few examples, like P. electobats have high interferon alpha basally, but we, for example, looked at the RNA cell pathway in bats versus humans, and it's really similar. So there probably must be important differences, but it's not so clear what they are.
Do you think there has to be an intermediate animal from bats to humans for SARS-CoV-2?
Okay, so I don't think there has to be one, because if there's some of the bat viruses, as Peters has shown, may be, and Ralph may be able to infect human cells. Mm-hmm. So I don't think it's 100%. I think probably there is an intermediate species, but I think it's possible that there isn't.
So do you think it could go right from bats to humans, right?
I mean, that's total speculation, but I think it's possible if it would have to adapt.
Yeah.
I'm sure.
You think we'll find out at some point in the future how it happened, the spillover?
I think so. I mean, we found out with SARS, it didn't seem that hard, that long. And MERS, of course, so MERS is really interesting to me. The camels, it's just sort of maintained in the camels. Yeah. It's different from SARS, where it seems probably was kind of briefly in the intermediate species.
And in fact, these other, the OC 43229 E. I'm going to remember those at some point. Some of those came from bats or rodents, right?
Well, OC43 supposedly is very closely related to MHV and BCV, the bovine virus, and probably from rodents. But 229E has there are 229E like alpha coronaviruses in bats, I believe.
Where are they located? Do you know? Are they North America or?
I don't know. I'm pretty sure there are alpha coronaviruses in bats in North America, but I don't know if they're the precursor to 229E.
So did you feel compelled to work on SARS-CoV-2?
Yeah.
Yeah.
Yes. Yes. We were already working on MERS, so we were all set up in BSL-3, and people in my lab were already working on these types of viruses. So and of course, I mean, we were the only ones in our institution, other than one other lab, that was approved to work in BSL-3, and we got the virus right away. And but we've done a lot of. We don't have a ton of data, but we've done a lot of just getting it going kind of thing and helping other people, like the clinical people, viruses.
So from a basic virology viewpoint, what are some of the interesting questions about the virus?
Well, one of the really interesting questions is, why is it different from SARS-1? Yeah. But I don't know that the genome doesn't tell us that. So I think it's going to be, the thing of this asymptomatic spreaders, that seems going to be really hard to figure out from the genomes.
Right.
I mean, I'm interested in how it interacts with its host cell and innate immune response. That's the first thing we want to look at, compared to, because MERS and MHV that we've worked on a lot, they really shut down interferon pathways. And actually, there's this interesting thing from Stanley Perlman's work and also from humans, that during early infection, there's really interferon can knock the virus down. It's therapeutic, whereas later in infection, it becomes pathogenic. that's part of the cytokine storm. So that's really curious because the virus itself shuts all that stuff down.
So I'm just waking up to this here. I hadn't really considered that SARS-1 and SARS-2, we know are actually quite different in their pathogenesis in terms of asymptomatic transmission versus more symptomatic transmission, mortality rate, et cetera. And yet their genomes are quite similar. But if you do that comparison and look at the genomes, it doesn't really, nothing jumps out at you that would explain those differences, right?
Exactly, yeah. So, I mean, there are differences in the accessory, about the accessory protein. So we have the replicase that's about 20 kilobases. We have the structural proteins that are all in the same order in all the viruses and all the genera. And then there are accessory proteins that are different for each either sublineage. So among beta coronaviruses, the lineage A, B, C, D are all different. So those are pretty much different, unique. But for SARS and SARS-2, they're very similar, although there are some differences that are come out. I don't know if it has anything to do with that.
Do you have the capability to, the way this is you can't do this. The way this is typically segregated in other systems is to do the appropriate genetics, right? Isolate the differences one at a time in a common backbone or something like that, and then look at an animal model and see which things contribute to the pathogenesis.
So yes, you can do that. Theoretically, it's going to be hard. So there are 2 reverse genetic systems already published for this for this virus. One is Volker Thiel has a really interesting, unique system where you assemble the genome in yeast, and so it's a very quick. We're going to try it. We just got it. You can assemble new viruses quickly, supposedly, at least in his hands. And then the other one's a more conventional cDNA clone from the people at UTMB. So you can make mutations, isolate them, and make viruses and put it in an animal model.
You would need appropriate animal models that can distinguish the pathogenesis of these two viruses. Does that exist?
Well, I don't know. There are models now that look pretty good for hamsters, ferrets, non-human primates. The mouse models are evolving because it doesn't naturally infect the mice, so you have to make transgenic mice or adapt the virus. So that's all happening now. Stanley's been working on that, and Perlman. I don't know that anyone's looked closely enough to see differences in these models yet, like in terms of asymptomatic spreading. That would be great if we could model that.
Susan, if you lined up all the SARS or all the coronaviruses, and then made a list of all the receptor molecules on the host cell surface, do they fall into groups? Because I noticed in your presentation, at least SARS-1 and SARS-2 use 2 different receptor molecules.
No, SARS-1 and SARS-2 use the same one. It's MERS that.
I'm sorry, I meant MERS. I did mean MERS.
So the answer is it seems really random to me.
Random.
Well, because SARS-1 and SARS-2 use ACE2, but so does NL63, one of the less pathogenic viruses. And it's an alpha and they're beta. So that seems really random to me. And then OC43 doesn't really have a distinct protein receptor. It's sugars. And let's see. Yeah, and MERS uses a completely different receptor. So I think receptor interaction is a little bit overemphasized in terms of pathogenesis because you have NL63 and SARS, and they're completely different.
And how do you make out of this data that's arising now with people that are suffering from hypertension that are receiving anti-hypertension drugs and they're dying like flies? And that seems to be the receptor molecule for the virus.
Yeah, I don't know. I don't know. I don't know but people are told not to stop taking their medicines, right?
Yeah.
You'll die of heart attack.
Yep.
Don't stop.
I don't know. I don't think
right. And there are
that,
to be clear, there are alternate medications. There are alternate medications that do not affect ACE2 expression. And I think the current thinking is that the issue is not so much the medication as it is the underlying heart disease.
So, Susan, you spent a lot of work on MHV in animal models, right? So, do you want to do animal with SARS-CoV-2?
I would like to.
Yeah.
I would like to. But unfortunately, our institution is just getting our ABSL3 facility functional, which is something I've been agitating for a long time. So finally now we're going to do it. Now with this disaster, we're going to do it. So we have a delay. But I can't tell you how many people in my institution want to use animals like for all kinds of things for SARS-CoV-2.
It seems like at every place, everybody wants to work on this virus now. How do you feel about that from the early days when no one cared about it, right?
Well, I think that people move in and people move out. And that certainly happened with SARS and MERS. And whether this is obviously the big one, whether people will stick with it, I don't know. And whether it probably depends on funding too, if people can get funded for it.
Yeah. So what do you think is the end game?
Vaccine.
Right.
Basically.
And speaking of that, what do you think of the prediction by the Washington swamp dwellers that we're going to have a vaccine in January?
I thought Fauci says a year at least.
Yeah, it seems like even that is wishful thinking. Yeah.
Well, the Moderna vaccine is in trial now, so if that should work,
I just did the count this morning. There are 8 different vaccines
in trial.
In trial.
Because I think there are like 50 different approaches. Yeah,
Yes.
in development.
In developments, right.
But you don't think we all have to stay home until the vaccine comes out, do you?
Me? I'm afraid to go out.
I can't.
I mean, I hope that we have some therapeutics, some drugs before that.
Sure.
Right? I mean, if remdesivir is going to be good.
Well, and also a combination of adequate testing, which we still haven't quite reached, and protective measures, and really, really critically, contact tracing. Those, even in the absence of effective therapies or a vaccine, we can do a tremendous amount to control subsequent flare-ups. And so it's conceivable, to me at least, that we could get back to some things opening under some conditions with restrictions and just vigilant tracking of cases, and everybody who had contact with them then gets quarantined, and we can try and shut things down that way. But that's not the ultimate end game, obviously, and as you just said, a vaccine is really
but if we have like a combination of an antiviral and an IL-6 antibody or something like that, maybe it would be less scary to leave your house.
So Ralph Barrick has said that in a couple of years, this will be another seasonal coronavirus. Because, right now the older people getting infected, that's the problem. And usually coronas infect young kids and they don't get very sick. What do you think about that? Well,
but some young people get pretty sick. And now I saw some reports about children getting some pretty bad syndromes.
You get the Kawasaki syndrome.
So, but even people like 40 years old are getting sick and dying. So I don't think it's only an old people's thing. And
I think Stanley told us that too.
Yeah, I think it's also true that we all know this, that we have no immunity at all. Like the only people that might have immunity are the survivors of the SARS epidemic. And I wonder whether they do. I haven't seen that anywhere.
Probably not too many of them, right?
No, they're 8,000.
Yeah.
But I mean, if we all get, if there were a vaccine, I think we'd all have some immunity, and then it would be more seasonal.
I know that you mostly focus on innate immunity, but do you have any thoughts about the persistence of the adaptive immune response to some of these coronaviruses and how that might influence vaccines or herd immunity?
So I don't have any hard data except just that generally people say that OC43, you can get multiple infections of it. But I don't think it means that you get it like a month later. I think it means a year later, but I think you still have some memory immunity. To me, it seems it would be really anti-dogmatic or really unusual if this virus, the immunity just went away completely. That doesn't make sense to me.
No, not really.
No.
Well, these we talked about this yesterday, but these recent supposed infections in South Korea within a week or two, it turns out that's just PCR
fragmentation,
pieces of RNA, no infectious virus.
Right.
I think, let me tell you, I really think that the PCR it sometimes is misleading because just from the samples we've been getting and trying to isolate virus from, I think you need a really high number of genomes to isolate a virus. And partially that's because of junk and fragments, but also these viruses have a really high particle to PFU ratio. At least I don't know about this one, but in general. So, like 10 genomes is probably not infectious.
Susan, this is on the nerdy side of a question, but you pointed out in your talk that the probes that are typically used in the PCR are going to have varying sensitivity because in a replicating virus, the messenger RNA for the N gene is going to be much, much more abundant than the RNA for the replicase. Right. Now, the kits that are out there, do they include both probes?
I think they do. I actually think they do.
And so, but when you get an answer, it doesn't tell you, or does it discriminate between the two probes?
I'm not sure. You probably just get a yes or no, but the lab may know. But this is one thing. Here's a nerdy thing too. Like when you take a sample out of the nose or something, is it cell associated virus you're looking for, or is it free virus? And that's really not clear to me. And how and
now they're using saliva, right?
That's what I hear. It's starting to be. First, I heard saliva had very little. Now they say saliva is better.
Yeah.
So saliva.
These scientists, you just can't trust them,
They spit and run.
That's right.
So in saliva, I don't know if it's cells or virus, or it's probably both.
That's a really interesting point.
Yeah.
Isn't there a lot of RNase in saliva? So
that's the other thing too. And they say there's nothing in urine, but maybe it has to do with the pH. I mean, some of these are not great environments for virus.
I mean, it seems that RNA is such a volatile or
fragile.
Labile. That's what I meant is labile. Of course, you have to autoclave all your glassware to make sure that the RNase is all dead. And here this virus is just laughing at all this,
But it's protected, right? It's inside of its membrane. And
no. So that's what I'm thinking, that saliva probably is virus rather than just RNA, naked RNA, right?
No, it wouldn't be naked RNA. Naked RNA would not survive. I agree.
There's also this phenomenon with RNA. Researchers, molecular biologists, think that RNA is this ridiculously fragile molecule that vanishes when you look at it. And I think a lot of that is because these molecular biologists are sitting in laboratories that are littered with RNase from all their mini frames. And RNase is a nice, durable enzyme that's just sitting around on your bench waiting to digest some RNA. Out in the actual world, outside the lab, I think RNA is a little more persistent.
My thesis advisor always told us it's on your fingertips. Watch out.
Yeah, we used to call it fingerase.
Yeah, right. But the PCR test does not require a 30 kilobase genome. It requires just enough to amplify that little piece. So it can be broken.
When I was at Rockefeller as a postdoc a long time ago, the first year I was there, I remember I was walking into Founders Hall. I don't know if you're familiar with that. That's the big building up the walkway. And some group got out of a van and got out this gigantic, what looked like a bunch of Tinker Toys. And they asked me if I could hold open the door while they walked in. Well, it turned out to be the model for the crystalline structure for RNase. And they were sharing it with the group at Rockefeller. And they both ended up getting the Nobel Prize for that. So RNase must be an easy enzyme, at least to isolate and to study in that sense.
You take a pancreas and you boil it in sulfuric acid. And the only thing that's left is ribonuclease. I'm not kidding.
No, I think he's right, because I remember they used to tell us that you had to dry heat glassware to really get rid of RNA cell. Like even autoclaving wouldn't do it completely.
Yeah, it also is one of the first molecules that was shown would refold from a denatured form. Okay?
Interesting. Yeah.
So,
that's right.
Hey, do you remember diethyl pyrocarbonate?
Yes.
Sure.
EPC.
That's what we used to use to inactivate it, and then you would autoclave to boil that off, I believe.
I think people still use it.
Oh, yeah.
Yeah, they do.
So why is RNase evolutionarily, why is it so stable?
Good question.
No answers. Yeah, I know. We have no answers.
So it's not only evolutionarily stable, it also has a relatively big impact on you as a researcher if it happens to get into your experiment.
Right.
Absolutely.
Maybe it was one of the first proteins to emerge after the RNA world.
Like a protection.
Or or in the RNA world.
Yeah.
Yeah, but there was probably RNA, then protein, and then DNA, right?
Sure.
So
Yeah, sure.
that could have been, and that may have been involved in protecting the RNA initially, and it evolved to do other things. Who knows, right?
Maybe it was the first predator.
It was the first
That's
predator.
what I was thinking.
The first predator, RNase. That's great. I want to eventually circle back to interferon, Susan, but one more thing. I wonder if you can explain cleavage of spike and what furin versus TMPRSS2 and other things do.
Oh, this is
Good
my favorite
question.
topic, actually, because I think there's a lot of misinformation, but just a lot of confusion about it. So some spikes have furin sites and some don't. If they have so during morphogenesis or assembly of the virus, or during assembly, or I should say processing of the spike, there's furin protease in around the Golgi and those intracellular membranes, and spikes that have furin can be cleaved there, so they're kind of primed for fusion. Well, I should back up for a second. So there has to be two cleavages for fusion, one between S1 and S2, and that's generally a furin site, and then another downstream fusion, another cleavage at S2, and that S2 cleavage reveals the fusion peptide that's required for fusion. And so those cleavages can happen in different places. They can happen on the membrane. They can happen during assembly, or they can happen in the endosome during entry. And so depending on where those cleavages happen, that kind of directs the virus into one path. There are two entry pathways, one through endosomes, which in that one, cathepsin is the cleaving protease. And the other one at the surface, it could be TMPRSS. That's where TMPRSS would be active. And I just saw a really interesting paper that said that showed that for SARS-CoV-2, you needed both furin. Furin would cut S1, S2, and TMPRSS would cut the S2 prime. So and this is also very, very cell type dependent. Like someone in my lab found for MHV, there's a zinc metalloprotease required on the membrane. So it's very complicated, but it clearly has to have those two cleavages, and the viruses that don't have the furin site have to have more cleavage upon entry, whereas the ones that have the furin site are kind of semi-activated already when they're made.
What did SARS-1 have? Did it have a furin site?
SARS-1 did not have a furin site, and that's why everybody was so hysterical over this fusion site, because SARS-1 and furin site, SARS-1 and all the SARS-related bat viruses do not have the furin site, whereas MERS and MHV do. So generally, the ones that are the lineage of SARS do not. This one does. That's why I said it's really interesting, and it could be a way to figure out where it came from.
Or you think that furin is essential to its pathogenesis in humans?
Probably not. I would guess it's not. Because it would just mean that would have to be cleaved by a different protease. Maybe it's going to be important. I would predict it's not.
Okay.
And this is the other thing that's kind of curious. If it goes in through the endosome, like hydrochloroquine is only targeting the endosomal site, and TMPRSS inhibitors only targeting the cell membrane site. So neither one of them should work completely unless the virus really, you know what I mean? They're kind of mutually exclusive in a way, or you'd have to use both of them. I'm just using hydrochloroquine as an example. I don't know that it works. I'm not endorsing it. That you'd have to kind of target both sites when you don't really know which pathway it's going to use, and it could use different pathways in different cells.
So both cleavage sites have to be cleaved for infectivity, and that can happen on the way in or the way out.
Right, or partially both. I think F2, the second cleavage, I think only happens on the way in.
Okay.
And then when it's on the when they're on the membrane, they're on the plasma membrane, that's how you get cell fusion if you have an activated. So generally, the spikes that have furin sites can activate cell-to-cell fusion, and the ones without don't. Like SARS-2, we're finding it has tons and tons of cell-to-cell fusion.
Okay. So Eric Schneider, right?
Snyder, yeah, SNIJ.
Yeah. So we did a paper of his a few weeks ago where he showed he was placking out SARS-CoV-2 in viro cells, and as you pass it, it loses the furin site.
Oh, I heard that too.
But I don't know what viro cells mean because they're Vervet, right? So
they're monkey cells.
Yeah. So
it means that you don't need the furin site in
monkey cells. Yeah.
It means that it's using another pathway. It's going through endosomes. I think is chloroquine active in that system? I'm not sure.
And Viros, I don't know. But it's
We found that it's much more fusogenic in human cells than in monkey cells. I don't know what that is either. And I don't know if it loses the furin site in human cells either.
That would be interesting to look at, right?
Yeah.
Or take it out and see if it still replicates.
I would bet it's going to still replicate. Right. I don't see how it wouldn't. Like let me just tell you an MHV story briefly. For MHV, there are strains that have very strong cleavage, like 100% cleaved spike, some that do not cleave at all, like 0, and some that are intermediate. And they're all completely pathogenic in different ways.
Mm-hmm.
So it's, yeah. And they're all more or less sensitive to something like a cathepsin inhibitor or something that keeps the pH high in the endosomes.
Yeah.
When you say pathogenic in different ways, what do you mean?
Well, they kind of had somewhat different tropisms. Like the virus that's really CNS tropic has completely cleaved spike, and it depends on a cellular, a membrane-associated protease for entry, and it's completely independent of the chloroquine pathway. But like MHV2, that's highly pathogenic in the liver, requires the endosomal pathway and isn't cleaved at all. But if you take this, but this is even more complicated, because if you take the spike from MHV2 and put it in the background of the more neurotropic virus, it will then infect the brain. So again, spike and spike entry is not the only determinant of tropism. It's a little complicated, but anyway.
Is SARS-CoV-2 neurotropic at all?
People say that it causes neurological symptoms. It certainly causes the loss of smell. So that's one thing I'm kind of interested in. But I don't know that we know that it actually gets in the brain.
Mm-hmm.
Ask Chris Cuomo; he'll tell you.
Well, what about SARS-1 and MERS? Do they get in the brain at all?
Not that I know of.
No.
I don't remember anything like that.
So, what cells do we know for sure in people does SARS-CoV-2 infect besides the respiratory epithelium?
Well, people are saying now perhaps macrophages, endothelial cells, maybe.
Mm-hmm.
Gut tract?
Yeah, maybe. Yeah.
Because there was some diarrhea associated with this for a while.
Not lymphocytes, though, right?
I don't think so, but I don't know for sure if anyone's looked carefully. And
what about kidney cells?
Kidney cells, yeah, probably.
Uh-huh.
I mean, ACE2 is more widespread than respiratory system.
Yes.
But again, it's not only the receptor.
Yeah.
Right.
All right. So let's talk a little bit about interferon. We'll make Brianne happier.
Yay!
Make me happy too.
So you did a lot of work with MHV and interferon antagonists. So let's start with these phosphodiesterases.
Okay.
And explain like what they are and how they work.
Okay. So, and again, these are not, the phosphodiesterases are found in the lineage A and lineage C beta coronaviruses. That means like MHV and MERS, but not SARS and SARS-CoV-2. And so what they do, they're really very specific. They're enzymes that cleave 2', 5' linkages, which is a really unusual linkage. The only thing we know that does is 2'5A is the activator of ribonuclease L. Mm-hmm. And also 2'5A linkages, I think, have some role in splicing. Yeah. So during infection, usually double-stranded RNA, which all RNA viruses produce, and even some DNA viruses produce, is recognized by a bunch of receptors, one of which is oligoadenylate synthetases. And when they recognize double-stranded RNA, they produce 2'5A, this small oligoadenylate. It's like 2 to 5 or something. And that activates ribonuclease L, which cleaves single-stranded RNA, viral and cellular, and that is antiviral because it kills RNA. It stops protein synthesis, causes apoptosis, and a whole bunch of stuff like that. And one of the things, so these viruses, MHV shows the most profound effect. They shut down that pathway completely. And for MHV, once that pathway shut down, the pathogenesis is completely apathogenic. The liver, for replication in the liver, but not in the brain. And so there's a whole lot of complicated stuff. But what I think is really interesting that came out of that whole study was that if you can shut down the MAVS interferon pathway, yet you still activate RNA cell. It's kind of an independent parallel pathway. So it's really important for viruses that shut down interferon, which a lot of viruses shut down to some extent or a large extent interferon production and signaling. So
I just love this, because if you were on the sort of joint chiefs of staff of coronavirus invading a cell, okay, and you looked at it, you'd say, look, we're going to have to deal with this RNA cell pathway that's dealing with double-stranded RNA and makes this weirdo molecule 2,5-A. And the best way to do that is going to be to take the double-stranded RNA out and destroy the 2,5-A. So you guys go off and make enzymes that'll do that. And that's what happened. It's amazing.
Right. And what's even more amazing, that pathway is really important because some work by Volker Thiel that we actually collaborated on too showed that if you knock out, there were actually two enzymes that MHV makes that attack this pathway. The other one is endonuclease ENDO-U, and ENDO-U is really interesting. It reduces, without telling you the mechanism, it reduces the amount of double-stranded RNA in the cell. So it knocks down all those pathways, all the double-stranded RNA pathways, RNA cell, interferon, and PKR. But that protein, which is in all of the coronaviruses, is not enough to really get rid of RNA cell, so that some of them have to make a whole separate protein to do that. I find that amazing, actually.
So
there's such redundancy.
That's the proofreading protein, right? That's doing.
No, that's XON. No, that's XON. It's XON and ENDO-U.
Okay, fine.
ENDO-U is not the one that does the 2' 5' cleavage.
No, that's 2,5. That's PDE or 2. That's NS2. It's not. See, that's an accessory protein. Okay. Whereas ENDO-U and XON are both replicase locus, are conserved among all the viruses.
Okay, so NS2 is the one that does the 2' 5'. Right. I'm thinking of fun stuff right now. So
Susan,
but in MERS it's NS4B. It's a different protein completely.
But the same activity, though. Right?
The same activity, but in a different part of the genome, which I find very curious.
So phosphodiesterase, if you take it out of MHV genome, what happens? Does the virus replicate?
It replicates in tissue culture, but cultured fibroblasts, transform cells, but it's completely dead in bone marrow derived macrophages or in vivo, but not in the brain. Okay, this is another really interesting thing. So the brain has really low interferon stimulating genes, basal ones. Right. Liver has really high ones. So you really don't activate RNA cell in the brain with MHV.
Right. Okay, makes sense, because you probably don't want to damage the brain, right?
Probably, yeah. It's like immune privilege.
Yeah, right. So this virus is debilitated. So this phosphodiesterase has no other function except to antagonize RNA cell, right?
We don't know. So a lot of this stuff we did with Bob Silverman's lab, who's probably done RNA cell as long as I've worked with coronaviruses. And he's very interested in finding other functions, but we don't know any right now.
Okay. And then the other one, the ENDO-U, is that what it was? Yeah, exoribonuclease, NSP14. If you take that out, does the virus still replicate?
ENDO-U is, just to be precise, is NSP15, and XON is NSP14. If you take out ENDO-U, you also activate RNA cell and you activate PKR. So an ENDO-U mutant will only replicate in like a cell devoid of both RNA cell and PKR. Whereas the NS2 mutant you only have to get rid of RNA cell.
All right, and SARS-CoV-2 has neither of these proteins, right?
When we searched databases, we couldn't find this is another curiosity, the only other virus that has a PDE, rotaviruses, completely unrelated, but a homologous protein. All right.
So does SARS-CoV-2 have to antagonize the RNA cell?
We're going to find out next week.
Okay.
Is there much interferon production? Or have you seen much interferon
production? We'll find out next week.
Okay.
That's exactly the experiments we're doing now. But we had to get the right cell to do it, because you can't do that experiment in a vero cell because they don't really have interferon.
Right.
Yeah.
So did I miss something? I probably missed a whole bunch, but did I miss the fact that this virus doesn't kill its cell? It buds out of the cell and just keeps producing, and the cell stays alive?
Not exactly, because if it produces syncytia, these huge multinucleated syncytia that eventually lift up off the dish, and it's not viable. No, but they do leak out of the cell. They don't lyse the cell. They
Right.
kind of come by like a reverse fusion
So
mechanism.
you can have a lot of cells in your body infected with this and still have functionality of those cells.
I don't think they function so well.
I'm just wondering where the cytokine storm comes from, because I always figured that was due to the lysis of the cells and all the debris, and then the cleanup, and then all of a sudden you've got this big mess. But that doesn't look.
Well, you're certainly going to have cell lysis and stuff like that. But like I said, I don't know. Syncytia, you don't. At least when we looked with MHV, we didn't see a whole lot of syncytia in vivo. It seems to be more an in vitro phenomenon. So I'm sure you're killing the cells. I mean. Yeah,
You're going to have a whole bunch of novel proteins showing up on the surface of these.
Yeah, well, I
infected cells.
thought about that too.
And that's going to set off the cellular immune response.
Yeah, and just things like the nucleic acid leading to, different types of immune responses as well.
I mean, the only thing on the surface is going to be the spike, but then when the cells lyse, everything's obviously going to be exposed.
So, Susan, aside from PDE, phosphodiesterase, and exoribonuclease, are there any other interferon antagonists in the MHB?
Yes. There's the I mean, not literally interferon antagonists, but the capping and the capping enzymes or prevent detection by like the MDA5 and RIG-I. These viruses are only detected by MDA5, which is kind of interesting too, not RIG-I. There's also NSP1 knocks down, well, not really knocks down interferon synthesis. Some of the NSP1s, they're a little bit different from virus to virus. There's also the macro domain. Mm-hmm. And there's also the ubiquitinase that's part of the papain-like protease polypeptide. Okay. So there's a lot. It really wants to shut down the host.
So the capping thing kind of blew by me there, and that interests me. Is it like make a chemically different cap or something?
No, just that it protects the viral RNA from.
Well, it probably makes it resistant to IFIT, which binds, modified unmethylated caps, and this should be normal, right?
Yes.
It's so interesting to me, at least, to hear about a virus an RNA virus that comes in with such an enormous array of armamentation against the cellular immune system, and I assume it's because coronaviruses, with their proofreading function, are able to have longer genomes than a lot of RNA viruses, and a lot of other RNA viruses are just they have a different strategy for dealing with this that doesn't involve this protein and that protein and this protein all interacting. This is almost like a DNA virus type of strategy.
Kind of.
Yeah. What I like to point out is that this means that there's a lot of potential targets for antivirals.
Mm-hmm.
Yay!
Absolutely.
Well, that was my point in my talk. There's like 10 enzymes or something. Yeah. The primase, there's a primase, a helicase.
And a lot of this stuff is heavily conserved among coronaviruses.
Yeah.
So you could, you could potentially come up with drugs that wouldn't just be useful. I mean, obviously, finding a drug that's useful against this virus is a pretty big deal right now. But 10 years from now, when something else emerges, you might be able to recycle the same thing.
Exactly. I mean, the remdesivir and the RDRP and the two proteases. I guess those are the ones that people talk about the most as targets. And they're really conserved among all the viruses.
Right.
Susan, how long is the replication cycle? Can you generalize to say that it's slower than smaller RNA viruses, or is that an overstatement?
No, I don't think you can. Like, for example, MHV in an L2 cell will be very quick.
Okay.
It depends on the virus and the cell type.
What do you mean, like 12 hours?
At the most.
Still, polio is faster.
Oh, shit.
Polio is faster than most.
Polio is like 1/4.
Oh, yeah. Well, my capsid can beat your capsid any day of the week.
Of course, Condit is going to tell us that his phages are 20 minutes.
Yes.
Right?
Well, that was a long time ago.
And by the way, vaccinia virus activates RNA cell really well, except when you take out E3L, it's crazy.
Yep.
So, Vincent, how long did it take you to synthesize poliovirus?
About a year.
Right. That's a long replication cycle.
That's just me. That's because I was slow.
It took so many years, and with Ralph and everybody helping him to make those first clones. It took forever.
Yes. Yeah. I was lucky I picked an easy virus. Yeah. So you expect that SARS-CoV-2 is going to have interferon antagonists, right?
Yeah, I'm sure it does.
And
I mean, ORF6, for example, is in SARS-1 prevents STAT translocation to the nucleus. That's one thing we're interested in seeing if that's true.
I was wondering if you knew anything about ORF8, because it was deleted in SARS-1, and now in Singapore there's some isolates with it deleted.
Yeah, no, ORF8 started off as ORF8 in SARS and then became ORF8A and B by deletion of some, and then the larger deletion. I think it probably means it's not that important in humans. I'm just guessing. But
we don't have any idea of what it's doing, right?
I don't know what it does.
Okay.
Somebody might know, but I don't.
So you have this paper, which I was interested in because it includes Adar, right?
Adar, yeah, that's our other favorite.
Tell us an Adar story in coronavirus.
Okay, so Adar, in the absence of Adar...
Well, tell us what Adar is.
Okay, adenosine deaminase acting on RNA. It's an RNA editing enzyme. It edits A's to I's, inosine, and that causes mutation. But it also prevents double-stranded RNA from accumulating. So in the absence of Adar, double-stranded RNA accumulates under normal conditions in a normal cell, and it activates these pathways in the absence of viruses. So it's kind of like, and that causes diseases like Cardi-Gutierrez syndrome, which is a really horrible neurodevelopmental disease. Kids, it's very rare, it's genetic. So we try to understand what pathways are activated there and what causes cell death. And it turns out that, of course, RNA cell is activated quite well in the absence of Adar, and it leads to apoptosis. Yeah. So we got interested in that because of RNA cell and wondering which pathways were really the most destructive in the absence of Adar.
Yeah, and people who have mutations in the Adar gene, they may have interferonopathies, right?
That's what this disease is. It's an interferonopathy that upregulates interferon-stimulated genes, and that's like not good.
So is there anything else we should talk about that you're excited about?
That I'm excited about? I'm too tired to be excited. Now, for me, this has been a really interesting time because I've gotten to do some things that I never would have been asked to do before. Like I talked to the National Academy of Sciences annual meeting. I was on a panel with some pretty illustrious people. So it's kind of weird. I knew all this stuff like 6 months ago and nobody cared.
Right.
Exactly.
Yeah.
So do you see mostly working on SARS-CoV-2 going forward, or are you going to still do MHV?
I've been thinking about it. I think, well, not only in SARS-CoV-2, but coronaviruses. Comparing, we have 3 now in the lab, MHV, MERS, and this one. We can't work on SARS-CoV-1 because we don't have select agent permission. We could do that, I suppose. But yeah, I think this is really exciting. It's kind of rejuvenating me. As a virologist, yeah.
You were always rejuvenated.
Right.
So why is SARS-1 a select agent? Just was in that?
Totally random. I mean, it wasn't initially. And then one day, this is another crazy thing. One day, everyone who got SARS-1 from CDC, which was easy to get at the time, got a phone call and said, you either get select agent stuff or autoclave it. And so we autoclaved it.
Wow.
And it was so arbitrary because MERS never became one, and I'm told that SARS-2 won't be one. So it makes no sense. They should undo it, really.
Yes.
People tried to isolate the SARS-1 virus from bats again.
Not the SARS-1s, but there's SARS-1-like viruses in bats. Yeah.
Do you think we'll have another SARS-3 and 4 and 5 in the future?
I would guess, yeah. I mean, I think there might have been something between all of these epidemics that we didn't notice because it didn't kill people. Because it doesn't make
kill
sense.
enough people.
Like, why would it be so rare, right? Why would it only happen once every 10 years?
I guess you could do seroassays, but you wouldn't know what
you're looking for.
Like, I don't think any of the other viruses cross-react with SARS.
Yeah.
Well, and I mean, the general view of spillovers is that these types of events happen with various viruses, and probably coronaviruses are serial offenders in this respect. And either it doesn't get going or it doesn't spread person to person. And depending on where somebody dies and under what circumstances, it may have just not come to anyone's attention.
Yeah.
For sure.
Spillover and the amplification.
Yeah.
And this virus apparently lives longer on inanimate objects and on the fomites and stuff like this.
Okay, that's another bone I have to pick in a way, because when people do those experiments, you never know, are they starting with 10 to the 8th PFUs?
Right.
Seriously. Or 10 to the 3 PFUs. Like, how much virus are you actually, unless you actually sneeze on a surface, how much virus are you likely to be depositing on a surface?
Exactly.
And then there's a half-life. It's not like it's there and then it's gone.
Well, the thing I worry about is hospitals.
Yeah. Maybe.
Yeah, where there's a lot of virus around.
Right.
Susan, how many PFU of SARS-CoV-2 do you need to start an infection in a human?
I have no idea, but I'll tell you that, like, when we get a sample from the nose, most of them don't yield virus when we try to. So they might have, like, say, 10 to the 4 genome copies, and they may or may not easily get virus out of them.
Yeah, that's looking for your watch that you lost under the wrong street light. I think the spit test is much more likely to take a virus from the upper respiratory tract than the intranasal whatever.
Yeah, we also have what do they call ET and endotracheal aspirates too. We try to isolate from them, but they seem to have a lot of bacteria in them, and they're hard to.
Exactly.
But yeah, they may have more virus. I don't know what that number is.
I keep going back to, well, what I remember Christian Drosten saying, that if you have less than 100,000 genome copies, you don't have any infectious units. So
that's what I think that's right.
Yeah.
Because that word genome copy is probably not right? It's not a whole genome at all.
It could be.
And yet people are worried about breathing the same air that the jogger just ran by with you.
Yeah, I think that's a little bit over the top. I think that it might be whole genomes, because I do think there's really high particle to PFU ratios.
Yeah.
Even if there were whole genomes, it might still be okay.
Right. And we do know from just the epidemiology that there are situations like choir practice where people can expel huge amounts of, apparently, huge amounts of infectious virus, because we've got super spreader events to prove it. But that is probably dependent on phase of infection in that individual and so forth.
Yeah. I mean, if the clinicians sample the nose, randomly, it's not going to always be the same.
Right.
What's the infectious dose for MHV? How many PFU?
1.
Wow.
Well, it depends. Wait, I take that back. If you put 1 PFU in the brain, you'll get encephalitis.
Wow.
And it's an LD50, 1 in the brain. But in the liver, you need like thousands.
But that's injecting it directly into those places.
Yeah, so I have no idea what it is like oral fecal in the colony.
All right. Okay. Anything else before we wrap up from anyone? This has been the, the Joint Chiefs of Coronavirus. Yeah.
Exactly.
Coronavirus.
I feel like I'm the mother of coronaviruses these days.
The mother of all coronaviruses.
Right, that's how I feel.
I like that one. Put that down. Come on, somebody put that down.
But Susan,
don't put it down, please.
Have you been are you the longest working coronavirologist?
Actually, I was thinking about that. Kay Holmes just retired. She was certainly before me, and I'm trying to think if there are any other ones that are still active. That
what about
Julian? No, Julian started before me. Julian Leibowitz. Stanley was after me. Ralph was a postdoc when I started my lab. Yeah, and Mark Dennison worked with me for years. He was at Jefferson, and we had joint lab meetings for years.
Wow. I'd forgotten about that.
Yeah, we were pals.
Very cool. I want to make 2 public service announcements. Actually, Kathy, you're going to make one, right?
Sure. So we got this announcement from the American Lung Association. They're accepting applications for COVID-19 and emerging respiratory viruses research, $100,000 per year for up to 2 years. The deadline is May 30th, so you have to get going on that. It's addressing clinical, basic, translational, and population health questions, everything from basic biology to epidemiology, but not just COVID-19 related. It could be other respiratory viruses as well.
Good.
Okay, there you go. All right. The other thing I wanted to point out is that Kiki has been summarizing our Friday TWiVs, which just started. And so there'll be a link to those in the show notes called Kiki's Comments. And Kiki's a former student of mine, and she asked not too long ago how she could help. And I just gave her a second thing, and that's this. She's happy to do it. So they're bullet point summaries for people who, for some reason, don't want to listen to the whole thing. I don't know why you'd ever want to not listen, but
You should call it Kiki's Summary Service.
Yeah.
Kiki's Summary Service. Yes, that's great.
They're
And
really good summaries.
One
like
more
this?
comment, Vincent. We have that trainee in common who's now, I think, very active in this.
Oh, yeah, Scott Hughes, right?
That's right.
Yes. So he was a student with you, right? And then he came and did a postdoc, and he's working for the New York City Department of Health, right?
Yeah, he's pretty high up in that organization. Yeah,
Wow.
Disaster preparedness is his thing.
All right.
Excellent.
Hey,
I
Scott.
haven't talked to him recently, but I'm sure he's busy.
Oh, yeah.
Yes. Well, many people are. All right. That is TWiV 609, microbe.tv/TWiV for the show notes. And someone asked me if the show note meant the transcript. No, it's not a transcript, but we have links to articles and links to the letters when we read letters and Kiki's comments. So those are at microbe.tv/TWiV. You can listen on any podcast player, of course. Send us your questions and comments, TWiV@microbe.tv. And if you like what we do, microbe.tv/contribute, you could donate a little bit of money. Our guest today from Philadelphia, University of Pennsylvania, Susan Weiss. Thanks for joining us.
Thank you. It was fun.
Susan, you have a coronavirus institute. Is that what it's called?
Well, we have a new center for the research on coronaviruses and other emerging pathogens.
How new is that?
Like a month or two.
Okay.
It's an emerging institute.
It's an emerging institute. Very good.
That's right.
It just started. It was really in response to this.
Right.
Yeah, it's very timely, of course. All right, thanks for joining us. It was really cool.
Yeah, thanks.
Thank you.
Dixon Despommier is at trichinella.org and thelivingriver.org. Thank you, Dixon.
Thank you, Vincent, and thank you, Susan, for sharing all your experiences with us. And I learned a lot. Thank you so much.
Thank you.
Rich Condit's an emeritus professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure thing. Always a good time.
You going to the store tomorrow, Rich?
We picked up groceries yesterday. So that's right. Texas is open, isn't it?
That's
probably,
right.
Probably
Gotta go to the beach?
not. I've been dying to go to Lowe's and pick up some mulch and some topsoil. So I may yet venture out. We'll see.
Mulch and topsoil. Very exciting.
I want to go out and play in the dirt, man.
A Home Depot, at least, was considered essential in Massachusetts, so they've been open the whole time.
Oh, they're open here, too.
Yeah.
Lowe's has been open, yeah.
Kathy Spindler is at the University of Michigan in Ann Arbor. Thanks, Kathy.
Thanks. This is a lot of fun, and it was really great to have you, Susan. Thanks for doing this.
Oh, thank you. Nice to talk to you again.
Yeah.
Brianne Barker's over at Drew University on Twitter, BioProf Barker. Thanks, Brianne.
Thank you. Susan, this was really fantastic. Thank you so much. I have a feeling I'm going to be trying to fit in some reading about phosphodiesterase
Good.
this weekend amidst my grading. And thanks. It was great to be here. And it's finally sunny.
Yeah, the sun did come out.
Well, here it's getting cloudy now.
It's going down the northeast corridor. Brianne, do you teach in the summer?
I do not teach in the summer. I may have some students working in the lab depending on openings for the summer, and I'm actually on sabbatical in the fall, so I am done teaching for 2020. Just grading.
Cool.
You're going to go someplace?
Well, right now, I just hope that I could go to my lab.
Right. Oh, I hear you. I hear you.
I'm teaching a summer virology course at Columbia.
Oh, very cool.
Yeah, they asked me, they said, well, we're not having summer classes because we can't, but we're going to have an online summer. So they asked me to teach it 6 weeks, 4 1.5 lectures a week.
Wow.
1.5 hour lectures. Oh boy. But I just taught it, so it should be okay. Alan Dove's over at turbidplaque.com on Twitter. Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure. And Susan, great to meet you.
Oh, you too.
I'm Vincent Racaniello. You can find me at virology.blog. I want to thank ASV and ASM for their support of TWiV and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.